So our video is going to be talking about DNA sequencing by reversible DYE Terminator Chemistry. We have three main talking points.
The first is background and history of the overall topic.
The second one is demonstrating through chemistry and methods.
The third one is the disadvantage and advantages compared to other technologies. Let's get started!
Frederick Sanger, also known as the father of DNA, is a British biochemist who is known for studying the composition of the insulin molecule, which he won a Nobel Prize for in 1958. He is also known for the development of the method Sanger sequencing. So, what is Sanger sequencing, you might ask? Well, it's basically a method of DNA sequencing that was developed in 1977, in which the DNA must be amplified, then heated to denature the DNA. That way, it will produce a complementary strand as well as the template strand that is used, as you can see in the video. You see we have the template strand, and the first step is to then add primer. After, you will then disperse the primed DNA into 4 different reaction tubes. We then add DNA polymerase into all 4 tubes, as well as DNTPs, in which they have 4. After, only one type of DDNTP is added to each reaction tube.
Before sequencing, we have to purify our DNA sample. The first step is lysis, which is the breaking open of the nucleus to release the DNA. The next step is to precipitate the DNA. After breaking DNA free, it is mixed with other cellular debris. Precipitation separates the DNA from the debris. Then, Na plus ions, or sodium, make the DNA more stable and less water soluble. The alcohol is then added to separate the DNA out of the liquid. The third step is purification. Once the DNA is separated from the solution, it is rinsed with alcohol to remove any other unwanted material or debris that could skew the reading. Now we can redissolve our purified DNA and begin the real fun. The first step in prep is to have a purified sample of DNA. Next is tagmentation. This has to deal with transposons and transposase. Transposase is an enzyme that binds to the end of the transposon and creates a reaction of movement to another part of the genome. The 2 transposons fragment and tag the input data with adapters at the same time. Adapters are then ligated, and reduced cycle amplification adds additional sequences that allow binding interactions to occur. The green and pink are sequencing primer binding sites. The red are indices. The yellow and green ends on the primers are regions that complement flow cell oligo.
Cluster generation. Clustering is a process where each fragment molecule is isothermally amplified. A flow cell is a glass slide with channels that are coated with 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos. This oligos is complementary to the adapter region on one of the fragment strands. Next, a polymerase creates a replication of the hybridized fragment. Then the double-stranded molecule is broken apart and the original hybridized strand is washed away. Next, the strands are conally amplified through bridge amplification. When this happens, the strand folds over creating a bridge and the adapter region hybridizes to the second oligo. Again, polymerases generate the complementary strand, making a double-stranded bridge. Next, the bridge is denatured, resulting in forward and reverse strand into two single-stranded copies that are attached to the flow cell. From the reverse, the bridge amplification process is repeated over and over again at the same time. After total bridge amplification of millions of strands, the reverse strands are cleared and washed off, leaving only the forward strands. The three ends blocked to prevent further unwanted priming. Sequencing
Begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 nucleotides that are tagged with fluorescence fight for the addition to the growing chain. Only one is incorporated based on the sequence of the template. Then, nucleotides are excited by a flash of light and a signal is established. This process is called sequencing by synthesis. The number of cycles determines the length of the read. After completion of the first read, the read product is washed away. Then, the index 1 read primer is introduced and hybridized to the template. The read product is then generated similar to the first read. The read product is removed and washed away.
Data analysis. Sequences are separated into groups based on similarities and unique indices. For each sample, strands with similar base cells are clustered. Forward and reverse sequences are paired, creating contiguous sequences, are compared to the reference genome for variant identification.
Okay, so now let's talk about the advantages and disadvantages of both the first and second generation of sequencing techniques. Okay, so let's start with the first generation, of course. So the main advantage of the first generation is mainly that it's what started it all. Without Sanders and his colleagues, his homies, we wouldn't be where we are today. We wouldn't be up to this point. So as far as first generation sequencing goes, it can sequence many identical molecules and it also sequences using gel. But when you compare the two, when you compare the first generation with the second and third generation, you'll find that more advantages would include the fact that for the first generations you have fewer preparation steps, while as in the second generation and the newer generations you have a lot more steps and they're more complicated. So normally in terms of the Sanders method, it's more impractical to use this method when doing large projects, like for example what we're doing, the Syntomos microbiome project. It wouldn't make much sense to do it because in the long run it'll be a lot more expensive than if we choose to do second generation techniques. Advantages of the Sanders method and the first generation sequencing methods would include number one, poor quality. So in the first sets of bases, it would have poor quality because of primer binding. So you won't, in the gel, it wouldn't show up and it wouldn't amplify. The quality of the sequencing also starts to deteriorate after about 700 bases, which all in all means you won't get as much data out of it. So because of this, you'll have to get more samples to analyze in order to get more data. This isn't exactly a bad thing, especially back then. It's what they had and it was a step ahead, but you'll just have to do a lot more work and it'll include a lot more labor. And you also have to take into consideration the fact the amount of mistakes you'll make and the amount of samples you'll contaminate just from living and breathing, basically. And with it being so costly and so expensive, you have to be more cautious and more careful around it. Because of the second generation sequencing would include the fact that it allows you to reduce your sample size, which means that you will have less samples. It also can sequence up to millions of amplified molecules per run, which is a definite improvement from the previous generation, in which you can only get maybe one read from one sample. So for this generation, you can get thousands or millions of reads from just one sample. It also takes less time to do this. As you can tell, the newer generations are a lot better than the previous generations. Why? Because number one, they're less work. Number two, they take less time. Number three, they produce a lot more data. So basically, you get more for less. Definite improvement. It's also better because with better technology, you can do more. For example, you can do metagenomics. You can do larger projects like the Earth Microbiome Project. You can also discover mutations. You can sequence a whole human genome. There's a lot more that you can do.
Hey guys! So, our video is going to be talking about DNA sequencing by reversible DYE Terminator Chemistry.
Let's do that over.
Hey guys! So, our video is going to be talking about DNA sequencing by reversible DEY Terminator Chemistry. We have three main talking points.
No, we can't do that because I.
Don't forget to like and subscribe.
No.
Like and subscribe.
No, we just no. See, now we got. Now I mess up.